Hello, everyone. Welcome to today's surprise webinar, Biocatalysis, Sustainable Innovations in Catalysis for a Greener Future. I am Xiao Qiao. I'm a scientific editor at Catalysis Cell Press, and I'm very delighted to serve as your moderator today, together with my colleagues from Circularity and Cell Reports Physical Science. As biocatalysis is rapidly reshaping the frontiers of modern chemical synthesis by offering sustainable, highly selective, and energy-efficient pathways, now further empowered by AI-accelerated enzyme discovery and optimization, the field is opening for transformative opportunities across chemistry and biotechnology. It is our great honor today to be joined by an exceptional panel of leaders whose pioneering work continues to expand what biocatalysis can achieve. We have Professor Shelley Mentier, Professor Dunmin Zhu, and Professor Huimin Zhao. Their insights will help illuminate both the current landscape and the exciting directions ahead. We hope this session will inspire new ideas and conversations. Please feel free to submit your questions at any time. We will address them during the live Q&A. With that, it is my great pleasure to introduce our first speaker of today, Professor Shelley Mentier from the Missouri University of Science and Technology, where she serves as director of the Kalmar Institute Center for Resource Sustainability and the director of the NSF Center for Synthetic Organic Electrochemistry. Professor Mentier has published nearly 500 research articles and has received numerous awards recognizing her seminal contributions. Today, she will share some of the most exciting recent discoveries from her laboratory. So, Shelley, the floor is yours.
Thank you.
Yeah. So I'll stop sharing here.
Work on opening my slides. We'll give it a second for them to come up. Hopefully. So thanks for the very kind invitation to present today. I'm an electroanalytical chemist who has used biocatalysis throughout my career. As an electroanalytical chemist, you might think that today I'm going to talk about biosensors because I think biosensors are kind of the classic example of where an analytical chemist combines electricity or electrochemistry and biocatalysis. But today I'm going to tell you a couple of stories in other areas of electrosynthesis. Specifically, maybe if I can get my slides to work. Specifically, two areas. One focused on using biocatalysts to generate electricity. So basically energy conversion or energy storage devices. If the biocatalyst is an enzyme, we call that an enzymatic fuel cell. If the biocatalyst is a microbe, we call it a microbial fuel cell. But basically using either a protein catalyst or a whole living organism catalyst to actually generate electricity from biological fuels or from a organic molecule. The second side of it is using electricity to actually generate molecules of interest. And so these would be sort of electrochemical synthesis systems, or what we call electrosynthesis. So with an enzyme, we call that enzymatic electrosynthesis. With a microbe, we call that microbial electrosynthesis. And I'm going to talk about both of these today because they've been areas of interest in my group for quite some time, but with a focus, a biocatalysis focus today on enzymes and not necessarily on the microbial side of things, although we work a great deal in the microbial side. Having a little challenge getting it to go to the next slide. So on the energy side of things, if you think about sort of traditional fuel cells, the most common fuel cell is the hydrogen-oxygen fuel cell, and it basically oxidizes the fuel hydrogen, separates it into electrons and protons. The electrons go through the wire and do work. The protons cross the membrane, react with oxygen from the air to form water, and that's the reason they're considered green and clean because they're only producing water. And this system works really good if you are talking about simple molecules like hydrogen, methane, methanol, but we don't really have the ability to develop traditional electrocatalytic materials that break carbon-carbon bonds effectively. So if you want to work with a fuel like ethanol that has a carbon-carbon bond, you're going to have challenges with traditional fuel cell chemistry. So what my group does is basically use biocatalysis and the ability of sort of metabolic pathways to do deep oxidation of fuels and sort of interfacing those enzyme cascades with electrodes for energy conversion devices. An example I'm going to show you today is the Krebs cycle or the citric acid cycle. So you can think about this as a metabolic pathway in the mitochondria of a living cell that has a whole cascade of oxidoreductase enzymes. So those are the bright red enzymes and corresponding non-dehydrogenase enzymes that will completely oxidize pyruvate to produce carbon dioxide through both oxidation and breaking chemical bonds. At the point in time we got interested in biofuel cells, there were pyruvate biofuel cells out there that had used pyruvate dehydrogenase, but we were interested in sort of seeing whether or not we could use these catalytic cascades or deep oxidation to be able to produce more electrochemical energy and to produce that electrochemical energy faster. If we look at what a fuel cell typically looks like, you typically will have a oxygen reduction side that's going to basically take oxygen from the air. And then you have an anode side where we can actually titrate the proteins of the citric acid cycle to actually look at their electrochemical performance. And if we do that, next slide. Maybe I can get it to work. If we do that, you can see that what happens is as we add additional dehydrogenase enzymes and their corresponding non-dehydrogenase enzymes, we go from having a small amount of current that you see there in pink to a little bit larger in blue. But what you expect to see is kind of a doubling, tripling, quadrupling of the current and the power that you get out of the electronic device. And it's not until we actually have all of the enzymes of the fuel cell, we vent CO2, that we see this sort of large increase in current density and power density. And that's because any metabolic pathway, any natural metabolic pathway is going to have metabolic control within it. And so if we're actually not putting all the enzymes down to be able to completely oxidize, we start to build up intermediates. Those intermediates actually inhibit other enzymes of the pathway and result in us having not as much sort of current and power as we might want to be able to power whatever device we're powering with the fuel cell. We could take this same strategy and look at other fuels. And so if we go to the next slide. This is an example with lactate where we just took the exact same Krebs cycle cascade. We added lactate to it. So we added one more enzyme and you can see we get sort of similar current densities and similar power densities. And you might say, well, why is this important? And basically, if you think about an electronic device, electronic devices need to be able to convert energy quickly in some applications. So if you're simply sending a text message that is just text, no pictures, no videos, you can get away with a low power or a low current fuel cell. But if you're trying to FaceTime, you need to be able to convert those electrons convert that fuel to electricity as quickly as possible. Another example of this would be if you think about a transportation application, you can have a fuel cell that may be able to go 0 to 60 miles an hour in an hour and a half. Is not what you want. You want to be able to go 0 to 60 miles an hour in several seconds and not sort of extended periods of time. So that speed of energy conversion, or what is the power density of the device, device is important. The other thing that's important is actually the energy density of the device. And so if we look at the two examples I just showed you, I showed you pyruvate and lactate. Pyruvate is not very soluble in water. So if we take it all the way to its solubility and we only have pyruvate dehydrogenase, one enzyme, we will make a battery with about 70 watt hours per liter of energy density, where a typical sort of lithium ion battery would be in the kind of 250 to 300 watt hours per liter. If we do complete oxidation and we get every electron out of pyruvate, then we're able to get something that's sort of comparable to the lithium ion battery. But if we look at pyruvate, the second fuel that I showed you, it's much more soluble. So because it's much more soluble, we can pack much more energy into space, into the same sort of volume. And so even with just a single enzyme, we can get 500 watt hours per liter out of lactate as a fuel, and we can store 3,000 watt hours per liter out of complete oxidation. And I think this is important to point out because obviously we need high power in the form of being able to convert that chemical energy to electrical energy quickly, but we also need to be able to store a lot of power so that we don't have to be recharging our electronics all the time. So from that perspective, I think it's important to think about the fact that the cascades of enzymes or using biocatalysis with enzyme cascades gives us the ability to both increase the power and current that we get out of a system, but also the energy density. So that was just an example on the energy side, but roughly 7 or 8 years ago, I kind of expanded into organic synthesis. So at the time, Phil Barron and I had both just started as associate editors for JAX. We met at a meeting and he started quizzing me about sort of electrochemistry with his focus on kind of electrifying organic synthesis. And I realized that this might be a new opportunity for us to go into new areas. So as an electrochemist, I was really interested in sort of whether or not we could start a new NSF Center for Chemical Innovation. And so as we started to think about basically building a center that would be trying to make organic electrochemistry or organic electrosynthesis mainstream to the organic chemistry community, as an analytical chemist, I needed to go out and talk to the organic chemists and kind of figure out what their pain points are, what their challenges were, and why they were still not using electrochemistry. So I went out to both academic and industrial chemists and asked them, you know, why is it that you're using stoichiometric amounts of oxidizing agents or reducing agents and not using electricity or electrons? As an electric chemist, it's obvious to me I would never use stoichiometric amounts of oxidizing agents or reducing agents, but that's not so, I guess, obvious within the organic chemistry community. So I started asking them sort of what their issues were. And the same couple of things kept popping up over and over again. The first was that they didn't really have any formal training. They didn't know how to use a potentiostat. They didn't own a potentiostat. And so that's a perfect thing for a Center for Chemical Innovation to address. That's a workforce development issue. And so we have been working on, you know, developing curricula to try to bring electrosynthesis into the undergraduate curriculum so that people feel more in their comfort zone. And from an instrument perspective, Phil's group, as well as the rest of the center, were alpha testing and beta testing the electrosyn at the exact same time that I was interviewing all these people. So I knew that was coming down the pike and was sort of less concerned about addressing the no formal training issue. But the other issue that kept coming up over and over and over again is that electrochemistry is not known for its selectivity. So it is known for having a pretty narrow potential window and not being able to get selectivity. And so people kept, you know, especially in industry, comparing electrochemistry to photoredox and biocatalysis, and talking about sort of, you know, the challenges that electrochemistry had. And the one that just kept coming over and over again was, you know, if you're thinking about biocatalysts, they have this great selectivity, and electrochemistry can never get to that selectivity. And at that point, as a person who's combined biocatalysis and electrochemistry for years and years and years, I thought maybe this was really an opportunity for us. And so I'm going to tell you a couple stories there in bioelectric catalysis and using enzymes as catalysts for organic synthesis. One of those is kind of a focus on using the enzyme to give you selectivity, but sort of rather than having to add stoichiometric amounts of oxidizing agents or reducing agents, which are typically cofactors in the biocatalysis community, how we could eliminate the need for cofactors. And the other is kind of addressing some solubility issues that come up a lot when we're talking about organic electrosynthesis with enzymes. So the first story I'm going to tell you is about CH activation of inert alkanes. As you know, we're still a petroleum economy, and so a lot of our feedstock chemicals, a lot of our precursors are coming from the petroleum industry, coming from inert alkanes, and needing to be able to activate those inert alkanes selectively. And obviously, there's transition metal catalysts. There are heterogeneous catalysts. There's a variety of catalysts out there to do that. But if you look at over the last decade or so, there's been a lot of work in sort of using biocatalysis and its ability to have selectivity or to have selectivity engineered, like the cytochrome P450 example that's shown here, to be able to kind of functionalize that inert alkane selectively. And so from that perspective, you can think about a whole wide variety of biocatalysts that might help to address that. But most of those biocatalysts are going to be oxidoreductases. They're going to require some type of cofactor to function. And cofactor regeneration can be a challenging situation to do in an economical and a green fashion. So the story I'm going to tell you today is not about cytochrome P450. It's actually about alkane monooxygenase. The reason for this is that cytochrome P450s are relatively easy to interface with electrodes, but sort of non-heme containing metalloenzymes like this alkane monooxygenase are much more difficult. So it was kind of a challenging electrochemistry problem as well as an organic synthesis problem. So we started with this alkane monooxygenase. We like it, not just because it's challenging from being a non-heme iron oxidoreductase enzyme, but also because it has a broad substrate scope. So it doesn't just do hydroxylations, it does epoxidations, it does sulfoxidations, and it does demethylations. So it has quite a bit of activity from that perspective. So the first thing that you have to do in any of these sort of electrosynthesis applications is actually get a hold of the proteins. And we actually expressed, isolated, and purified all the proteins that are biologically responsible for doing this. So that's the alkane monooxygenase, a mediator protein called ALK-G, and reductase that is doing your cofactor regeneration. So expressed, isolated, and purified them, did enzyme assays to make sure that they were still active, and then went to actually see if we could put them into an electrochemical system where we did electrosynthesis. The electrosynthesis was actually to drive the system without having to add any cofactor, no NAD or NADPH needed, and therefore not having to add stoichiometric amounts of NAD or NADPH, NADH or NADPH, nor sort of adding any cofactor regeneration system. So essentially what we're doing is basically removing biology's cofactor regeneration system and feeding those electrons directly in from the electrode, from the cathode, instead of sort of through a cofactor mediation system. It turns out this is a complicated system, and because it has this mediator protein, ALK-G, and if that ALK-G sits on a bare carbon electrode, it will denature. We actually had to sort of add an electrocatalyst or a mediator, this toluidine blue, to the system. This is electropolymerizable so that we can get a nice coating on our carbon electrode to keep that ALK-G mediating. And at that point, we could use this cathode to actually look at a variety of different reactions. So this is a monooxygenase. It's oxygen dependent, so we don't really see much electrocatalysis or electrosynthesis until we add both the alkane, in this case octane, and the oxygen. But when we do that, we can actually look at the fact that it can do that hydroxylation. It actually can do are epoxidations, sulfoxidations, demethylations. So all of the chemistry, you keep all the selectivity and all the chemistry that the enzyme can do, but we're driving it with electrons from essentially renewable electricity rather than having to drive it with cofactor and cofactor regeneration systems. We can do that using a single enzyme. We can add enzyme cascades here so that we can do kind of one pot electrosynthesis of more value added products like amines. So we definitely have the possibility to work there. But we have challenges with this system in that some of these enzymes are oxygen dependent, and we have to actually control in that pot the time where we have a lot of oxygen and the time that we have no oxygen. The other story I want to tell you is just a really brief story. The example is the electrosynthesis of chiral beta hydroxynitriles, another system where you really want to get the selectivity out of the enzyme. But unfortunately, beta hydroxynitriles, their reactants, the intermediates, and the final nitrile products are not very soluble. And so solubility becomes a huge issue with biocatalysis. That's handled a lot outside of electrochemistry with co-solvents. Co-solvents actually don't work in electrosynthesis. And so we went to these biphasic systems where we have a second phase that's acting as a substrate reservoir and a product sink. And these systems do work. So we actually can sort of increase the production of our product by actually having a biphasic system where we have an organic phase and a water phase. We have to make sure that that organic phase keeps the enzyme active while also having a good partition number for the reactant, the product, and the substrate. But if we do that, you can see that we have significantly more product that's formed. So we keep that same selectivity, but we're able to produce significantly more products because we are able to improve the solubility. So hopefully in the last 22 minutes, I showed you that combining biocatalysis with electrochemistry, or what we call bioelectrocatalysis, does have applications not just to glucose sensors that you might be more familiar with from going to CVS or Walgreens and buying glucose test strips for diabetic patients. It also can have applications to energy conversion and electrosynthesis, but there are still things we have to think about. We have to think about the oxygen sensitivity of our enzymes. We have to think about immobilization, enzyme immobilization and stabilization for a lot of these applications. In electrosynthesis, we have to think about solubility and production rates, so it is a bit more complicated. So with that, I want to take just one last slide to acknowledge the NSF Center for Synthetic Organic Electrochemistry. Got me into the organic synthesis world and has been great fun to see how we can use our biocatalysts with electrosynthesis, as well as my research group, past and present, who have done all the work that I showed you today. And with that, I will turn it back over to the moderators.
Great, Shelley. Thank you so much for this fascinating and very insightful presentation about using biocatalysis and bioelectrocatalysis, which also segues very well into Duming's talk. So now I'd like to introduce Professor Duming Zhu very briefly so that I can leave more time for his excellent talk. Duming has a long and balanced background between academia and industry. He is currently the director of Tianjin Engineering Research Center of Biocatalytic Technology, affiliated with the Tianjin Institute of Industrial Biotechnology of the Chinese Academy of Sciences. And his research entails the catalytic use of enzymatic and biological tools to achieve formation of challenging chemical bonds and productions of industrially relevant molecules, from which field Duming will now talk about his biocatalytic synthesis today. So please, Professor Zhu.
Okay, thank you, Jack, for introduction. Let me see. So first, I will introduce a little bit about what we are doing at the TIP. So we did enzyme engineering and enzyme discovery, and then we studied the reaction mechanisms and developed new synthetic methodologies for chemical synthesis. So after that, we develop a process with the industry and collaborate with them, commercialize the technology. So we study different enzymatic reactions. So today I will talk about two topics. One is engineering of THDP dependent enzymes for the synthesis of high value added chemicals from formaldehyde and other aldehydes. The second topic is biotransformation of phytosterols to key intermediates for the production of steroid APIs. As we know, the utilization of carbon dioxide is a hot topic right now. Carbon dioxide can be first reduced to formic acid and then formaldehyde and then methanol. We are interested in the utilization of formaldehyde. So we are interested in the coupling of aldehydes and to synthesize alpha hydroxy ketones and then transform this ketone to different high value added chemicals. For the coupling of carbonyl compounds, there are two types of reactions. One is from alpha hydroxy ketones, another is from beta hydroxy ketones. These reactions proceed through different reaction mechanisms and also catalyzed by different enzymes. So there are some reports about the coupling of different aldehydes with formaldehyde. For the coupling of formaldehyde with other aldehydes, there are some challenges. The first one is low chemical selectivity. From the reaction mechanism, we can see from intermediate 3, when it reacts with another aldehyde, this aldehyde can be either formaldehyde or the other aldehyde, so can form two different products. Also, this kind of enzyme show low tolerance of aldehyde because the aldehyde functional group can react with amino group in the protein and deactivate the enzyme. Also, in some cases, also show the low activity and limited substrate scope. To do this, we first collect a library of THDP dependent enzymes. Right now, we have more than 200 wild type enzymes. We studied the catalytic properties of these enzymes. For example, we found this PABAL showed a relatively broad substrate scope. For the aliphatic aldehydes and real aliphatic aldehydes, these enzymes show high activity for the coupling with formaldehyde. But for substrate 7 and 8, that's benzoyl aldehyde and benzoyl acetaldehyde, this enzyme showed low activity. Also for this 3-hydroxy propanol coupling with formaldehyde, we cannot find any enzyme shows activity for these reactions. So we start with HEBIL. This enzyme shows good utilization level, also shows a relatively broad substrate scope. Start with this enzyme, we first find Newton 1, shows a little bit of activity, and then through several rounds of mutagenesis and screening, we got a few variants shows good activities. Here shows the kinetic parameters of these mutants. But when we carry out the reactions, use these mutants, we found M4 showed better performance. That's because M4 was more stable, showed higher TM, and we tested at a different substrate concentration. We can see M4 shows better performance, up to 300 millimole. The conversion reached 95%. For the reaction of formaldehyde with benzoyaldehyde, the enzyme shows low activities. Also show low chemoselectivity. When we use this reaction screening our library, we found this mutant enzyme shows a high activity for these reactions, but also shows low chemoselectivity because the product 1B was formed. So here we select the M3 as a starter enzyme to do direct revolution. So we found 3 mutants shows better chemoselectivity. We got the crystal structure of these mutants. We found the movement of L552 decreased the volume of the substrate binding site. Also reduced the substrate entrance channel. So we can see this compared to the M3 underscore 3. This substrate entrance channel is smaller than this one. We also calculated the volume of the substrate binding site. We can see the mutant M3 underscore 3 shows much smaller volume. So this enzyme, this mutant shows better chemoselectivity. Here from the reaction mechanism, we can use the distance from this carbonyl carbon to this cofactor carbon 2 as the measure for the activity. So we can see for the mutant M33, most of the distance is less than 4.5 Armstrong. So this mutant shows higher activity and better chemoselectivity. So we use this mutant synthesize some alpha hydroxy methyl ketones. This shows the 3 different mutants use the fed batch reaction mode. So we can see the substrate concentration can be increased to 700 millimole. After we got this THDP enzyme and can make different alpha hydroxy ketones, so then we focus on the transfer this alpha hydroxy ketone to different value-added compounds. So first we synthesize this 1,2 amino alcohols, use imine reductase. So the imine reductase can catalyze the reductive amination of different amines with different ketones. Synthesize these 1,2 amino alcohols. This is another example. This is at the preparative scale. Also, we can combine these 2 reactions in one pot, 2-step process to make this 1,2 amino alcohols. Below are some examples. We also start with this compound 29 and show 2 biochemical reactions and then 1 chemical reaction. We can make this important compound. We also use M4, combine these 3 reactions in one pot. So we can make this 1,2,4 butadiene. Also, we can combine this with transaminase to make this amino alcohols. Also, we can combine this carbon-carbon formation reaction with ketone reduction reactions in one pot concurrent process. So these 2 reactions can carry out in one step. So we can make these 1,2-diols. This is the S stereoisomer. When we choose another carbonyl reductase, we can get the RS enantiomer. So if we carry out this reaction, use a continuous flow addition method, So we can increase this substrate concentration to 900 millimole. So if we replace the carbonyl reductase with amine dehydrogenase, we can make this 1,2 amino alcohols. Yeah, this shows this enzyme also can suitable for make aliphatic beta amino alcohols. So we also combine this THDP dependent enzyme catalysis reaction with oxidation reaction and then reductive elimination reaction to synthesize amino acids. So here use oxidase to oxidize this this alcohol group to the carbonic acid group to synthesize alpha keto acids and then transform this alpha keto acid to alpha amino acids. We also got the mutant to catalyze the coupling of 3 formaldehyde molecules to form 1,3-dihydroxyacetone, and then coupled with a transaminase catalysis reaction to make this amino alcohols. So start with simple alcohols. We can first make the hydroxy ketones and then transform this compound to different value-added compounds like amino alcohols, diols, and also amino acids. So next, I will talk about the biotransformation of phytosterol to the intermediate for steroid drug synthesis. Steroid drug is a class of very important medicines. In the top 200 small molecule drugs, there are 12 steroid drugs, more than 8 billion annual sales. When we start this project in China, we use the dioxazine as a starting material to make steroid APIs. For the production of 1 ton dioxazine, we need 100 tons of diacetyl and also produce a lot of wastewater. From dioxazine to this intermediate also produce a lot of wastewater. Use toxic chromium oxidant. So we developed a process to transform phytosterols to different intermediates. The phytosterol is a byproduct from vegetable oil industry, so it's a redundant resource. Also, it's biorenewable. Based on the literature, also our own studies on the metabolism mechanism of phytosterols by mycobacterium strains. So we construct different strains for the synthesis of BA, AD, ADD, and 9-hydroxy-AD. For 9-hydroxy-ADD, this compound, this intermediate is not stable. It decomposed to this compound, HIP. This HIP then decomposed to carbon dioxide and water. So we identified the key enzymes and then construct 2 strains to make HIP and also HIL. After we got this strain, we can did the transformation. We can see from original strain, we got the crude product. It's a mixture of different compounds. So for the new strains, we can get this ADD, 9-OHAD, BA, HIP, and HIL. The content of this compound in the crude extracted from the fermentation mixture can be greater than 90%. So then it's easy for the downstream process. So we collaborate with a pharmaceutical company, commercialize this technology. So right now we can produce these 6 intermediates at a ton scale. So right now in China, for the synthesis of sterile APIs, it's gradually started from phytosterols to replace disgenin. So this slide shows the product line at the company. So this is this production line is automatic, so it can be controlled in the central control room. Because this is the first product line built by this company, it can be automatically controlled. So they built a nice central control room. So you can see this production process on the big screen. So right now we can use this production line to product these 6 intermediates. Just use different strain because the starting material process are almost the same. we also construct two strings to make compound 2E and 3D. That the compound 2E can be used to synthesize this API, dehydrogesterone. This API is very difficult to make because this 10-alpha methyl group. For the natural sterile compound, the methyl group at 10 is a 10 beta. So it's difficult to make this 10 alpha methyl group compound. This intermediates provide a new method to make this very important API. Finally, I will thank my collaborator and my team, and also thank the funding from the National Science Foundation and also the funding from the industry partners. Thank you for your attention.
All right. Thank you, Professor Zhu, for this wonderful talk. I already have a ton of questions listed up to ask you a little bit later, and I encourage everyone to also use the Q&A to write your own questions at any time, and we'll get to them at the end of the talks. And now I would like to move on to Professor Huiming Zhao. From Ming's last slides, we can already see that automated production will be very important for large-scale synthesis, and I'm glad to say that Huiming will be able to immediately tell us more about such automation effort. Professor Huiming Zhao also has an extensive prior experience in the biotech industry before returning to academia, and now at the University of Illinois at Urbana-Champaign, he is currently the Stephen L. Miller Chair of Chemical and Biomolecular Engineering and the Professor of Chemistry, Biochemistry, and Bioengineering. Huiming has led an illustrious career in synthetic biology and incorporation of machine learning and laboratory automation. And indeed, today he will give a lecture on expanding directed evolution to AI-driven automation. The floor is yours, Professor Zhao.
Okay, thanks for your nice introduction, and it's my great pleasure to attend this webinar. For this audience, I probably don't need too much introduction. Enzymes and microbial cell factories have been increasingly used to produce chemicals and fuels from renewable plant biomass. And this actually is a sustainable alternative to the chemical refinery that we are all familiar with. However, compared to chemical catalysis, enzyme catalysis still have many limitations. Particularly, they are not very stable and also not compatible with organic solvent. But the biggest limitation is really the reactivity is kind of very narrow, right? The type of reactions that the enzyme can catalyze are still very limited compared to the reactions that are catalyzed by chemical catalyst. On the other hand, the enzyme clearly has some advantages, such as the high selectivity, environmental friendliness, and also they are very easy to engineer compared to organic synthesis of the ligand used in organometalysis. And there are two actually big challenges in biocatalysis. One is how to efficiently create enzymes with the improved or new function, particularly new reactivity. And that is kind of the biggest challenge, I would say. And the second challenge is how to efficiently combine enzyme catalysis with chemical catalysis, because I don't think enzyme catalysis will ever replace a chemical catalysis. These are two kind of viable options, question is that how can we really take advantage of both type of catalysis to create more efficient synthetic routes? So direct evolution is one effective approach to address the challenges I just mentioned. And this is near dear to my heart because my PhD thesis was focused on direct evolution. So I developed a number of direct evolution tools for enzyme engineering, which also helped my former advisor, Francis Arnold, to get a Nobel Prize in 2018. And the concept of direct evolution is very simple. Essentially, starting from the parent gene, we just use a random gene search and recombination to create a library of variants, and then use high throughput screening or selection method to identify the improved variants, and then we'll repeat this and use the improved variants as the starting point for next round of improvement, and then we can repeat it again and again. And this is very effective for improving existing protein functions. However, it also has a major limitation. It's very hard. It's very difficult to use direct evolution to create enzymes with new functions or new reactivity, because if you can just do a simple calculation, right? The creation of new functions or reactivity typically requires multiple simultaneous mutations. So if we want to generate a library with 3 mutations, the possible library size is already more than 10 to the 10th, which exceeds any kind of a selection or screening method that we can develop. So clearly we cannot explore all those combinations to identify new functions. So a few years ago, not a few years ago, actually more than 20 years ago, we came up a strategy called in vitro coevolution. So essentially, we use some intermediate functions to bridge the gap between the wild type of function and the novel function that we want to create. And then we use direct evolution to improve the function of those intermediate functions. And then we can gradually bridge the gap. In that case study, we used the ligand bind domain of estrogen receptor as a test case, and we tried to create a new the ligand bind domain that can recognize a completely new ligand. In this case, it's the corticosterone. And if you look at the chemical structure of corticosterone and the original ligand, the estradiol, they differ in many places, right? And the protein didn't show any activity towards the corticosterone, but they did show some activity towards the intermediate compounds. So we can gradually improve the binding of the ligand bind domain towards those intermediate. Eventually, we can create a new ligand specificity. And this strategy actually was used by Merck and Codexis to engineer transaminase with a new subspecificity, which was incorporated in the synthesis of Geneva, which is anti-diabetes, which is a drug used for treatment of diabetes, right? So that is kind of a successful application of this strategy in industry. But this strategy still kind of has many limitations because the process is still kind of tedious. And also, in many cases, we don't know, what kind of intermediate functions we should use, in order to bridge the gap between the novel function and the wild type function. So about 10 years ago, we came up another new strategy is to combine photocatalysis with enzyme catalysis, because photocatalysis is very versatile. It involves free radicals, so you can react almost all kinds of functional groups, but it doesn't have high selectivity. While for enzyme catalysis, we know it has a very high selectivity, but it doesn't have a broader reactivity, right? But if we can use light to create the radicals in the cofactors that the enzyme use, and then we use the active site of the enzyme to control the selectivity so that we can achieve both high selectivity and broader reactivity, right? So that is kind of the simple idea. And actually it worked very well. we initially demonstrated that we could repurpose the ing reductase that use the FAD as a cofactor, and we can generate the radical on that FAD cofactor, and then use this artificial photoenzyme to catalyze the coupling reaction between the olefin and this carbonyl substrate. Right. So as I shown here, we can do the intermolecular radical hydroalkylation reaction, and also achieve the intermolecular radical conjugate addition. And more recently, we show we can introduce the fluorine motifs into the target product. All these reactions involve the carbon-carbon coupling reaction, and the product that we can produce are highly enantiomer pure, right? So that's something that you cannot achieve using chemical catalysis readily, but for enzymes, we can do that. And also, in addition to the carbon-carbon coupling reaction, we can catalyze the carbon-nitrogen coupling reaction as well to achieve the radical hydroamination reaction. And all these reactions actually follow the same kind of a mechanism. So we basically use the light to create the radical on the cofactor, and then use the light to create the radical in one of the substrate, right? And then the radical substrate can react with the olefin substrate to form this prochiral compound. And then the enzyme active site controls the hydrogen atom transfer in an enantiomer pure, in enantiomer selective manner to achieve the high selectivity. And here I just want to show the fluorination reaction briefly. In nature, there's only one class of enzyme, so-called fluorinase, that can catalyze direct fluorination. And but the subspecificity of the enzymes are very limited because they all use a kind of a same they all use the same as the substrate, right? So you really cannot change the substrate specificity that much. And also we know that the incorporation of fluorine can really change the property of the molecules, right? Almost 30% of the pharmaceuticals or the agricultural chemicals have fluorine in it. Yeah. And so there's a huge demand, to create a new, like a fluorinated chemistry, right? And our idea is to, as I mentioned in the previous slides, we actually add a leaving group to the fluorine, and then create the radical, and then we introduce another olefin substrate, and then we actually use the enzyme to catalyze this coupling reaction on the light, and we can create a wide variety of products with fluorine that shows high selectivity. And this reaction is kind of unusual, so we tried many different fluorinated agents, and we found that the trifluorine iodoethane, and this fluorinating agent works well. At least it can achieve high selectivity, but the yield is very low. And also we made sure that this is an enzymatic reaction, right? It doesn't, it's not like a spontaneous reaction. So we did some control studies to make sure it is indeed an enzymatic reaction. And we also tried many different substrates to show the broader scope. But we also explored this reaction at a different remote site. We can achieve the selectivity at the beta or gamma or even delta position. So at the delta position, I'm not aware of any chemical catalyst can achieve selectivity. But with the enzymes, because the binding pocket is pretty big, we could actually catalyze the enantiomer the stereo to achieve the selectivity at the remote site. But in order to do that, I think the wild type enzyme is also not a very effective. So we also have to use direct evolution to further improve the selectivity. Right. So we have to introduce mutations and then find look for the mutants that can improve the selectivity. And in addition, we did some computational studies as well, try to really understand what determines the selectivity, right? Why it produce, why the enzyme has a preference for S product rather than R product, and also we identify the residues that are responsible for the stereochemical control. So photoenzymes clearly, they are very versatile. They can catalyze many different type of reactions, but none of them is really used in industry, right? At least right now. I think one major limitation is that the catalytic efficiency of these enzymes are very low, and so you have to use lots of enzymes in the reaction, which made the whole process not economical. So that's why in the past year, we try to come up a new strategy to address this challenge. Our idea is to incorporate the artificial photoenzyme in cellular metabolism, and we want use the whole cells, right, to produce the two substrates used by the photoenzyme, and also use glucose as the feedstock, right, the substrate, because the glucose is very cheap, compared to any chemicals we use in catalysis. So as a proof of concept, we engineered a cell to produce this olefin, right, and then also produce this substrate, and then we have an engineered photoenzyme that can catalyze this coupling reaction on the light to produce this bioactive compound. And so we did a lot of enzyme engineering, because even we incorporate the photoenzyme into cellular metabolism, its activity is still very low. So we did a direct evolution study to improve the overall activity of the enzyme, and also did pathway engineering to generate the two two substrates, and then we have this engineered cell. Or you can think it is more like a programmable catalyst, right? It's basically we program everything. We program the substrate, program the catalyst, and then we only use the glucose as the feedstock to produce the catalyst and substrate and the product. And when we did the fermentation study, we found that it can produce like 0.6 gram per liter. And it is not a very high titer, but at least it shows demonstrate the proof of concept, right? It is possible to incorporate engineer the photoenzymes into cellular metabolism, and this will open up the product a lot of opportunities, right? It will basically now enable us to produce many compounds that cannot be produced using natural enzymes or even chemically. And now I would like to switch the gear. I want to talk about how actually we can use AI and robotics to accelerate the enzyme engineering effort. I already explained the principle of direct evolution, but the new paradigm we try to pursue or establish is so-called autonomous experimentation. So in this paradigm, we use AI tools like generative AI tools or large language models to make predictions on the enzymes that we should create. And then we use robotics to perform all the experiments, and then you also use the supervised machine learning model to predict the combination of mutations that can further improve the protein function, and then we can do it iteratively. So the principle is similar to direct evolution, except that we use AI to make predictions and guide the experience. And also all the experience will be done by robotics rather than a human researcher. And I think that the power of both direct evolution and AI-driven autonomous expression depends on the iterative cycle, right? Is we have to accumulate those mutations very quickly. And I started this effort actually more than 10 years ago. At that time, I was inspired by development of a computer on our campus. More than 70 years ago, a group of physicists on our campus designed one of the first computers called Illinois Automatic Computer 1, or ILLINAC. And it consists of almost 3,000 vacuum tube, weighted about 5 tons, but the computing power of that computer already exceeded all the computers in the Bell Labs combined. And so about 10 years ago, we built the integrated robotic system called the Illinois Biological Foundry for Advanced Biomanufacturing, or iBioFab. So just like a computer is for computing, we use the iBioFab or Biofoundry for rapid prototyping of biological systems. And this effort led to the formation of Global Biofoundry Alliance in 2019, which currently consists of more than 40 members around the world, and also many actually in industry. And it also led to the formation of the NSF Biofoundry last year, which I will talk about in a minute. So our idea is to really integrate a lot of instruments into a large robotic system, as shown in the video. The robotic arm will take a 96-well plate, from one instrument to another, right? And there are more than 20 instruments on the platform. So you can think of this as automated lab. And we use this lab to kind of perform all kinds of synthetic biology experiments. So we break those complex workflows for protein engineering, metabolic engineering, or just library screening into process modules, which will be in turn broken down into unit operations. And then we will program those unit operations to create custom designed workflows for different applications. And particularly with the incorporation of AI, we could actually achieve autonomous experimentation. And we started this effort in 2013 with the grant from Kava Trust, and then obtained the grants from various funding agencies, including DARPA, NIH, NSF, DOE. So basically, further develop the technologies for the Biofoundry, but also explore the applications of Biofoundry. So in 2020, we built the second generation of Biofoundry. And then just a month ago, we had a ribbon cutting for our third generation Biofoundry called iBioFoundry 1 with the funding from NSF. So we used this Biofoundry to achieve autonomous experimentation a few years ago. And this is a proof of concept study. We tried to use multiple enzymes to synthesize an important chemical called lycopene, and we express those enzymes in the whole cells. But we need to balance the expression of these enzymes to achieve high yield. So what we did is that we use the robotic system to construct a very small library of pathway variants that contain promoters with different strengths for the 3 structure genes. And then we use the robotics to perform all the experiments, right? And also quantify the product. And then we use a very simple AI tool called Bayesian optimization to analyze the data to make predictions on the combination of promoters that can further lead to high that can improve the production of lycopene, and then we can do it iteratively. So we only made 46 variants in each round, right? And basically here I show the workflow. We constructed the plasmid that contains those pathway variants and then transform them into E. coli for production of the lycopene. And then we use the plate reader to quantify the lycopene production, and then use the AI to make predictions, and then we can do it iteratively. We repeat the 3 rounds, and in total, we designed a construct like 138 pathway variants, which is less than 1% of all the total possible combinations, right? So the take-home message is that with this AI-driven pathway optimization algorithm workflow, we can significantly reduce the experimental effort. We can find the best optimized pathway very quickly. And a similar approach now has also been used for chemical synthesis or material discovery as well. So, and this proof of concept study helped me to obtain a grant from NSF in 2020 to establish one of the first 7 AI institutes in the United States called the Molecular Maker Lab Institute, or MMLI. So in this institute, we try to develop a new AI, new chemistry, and biochemistry to radically accelerate and democratize molecular synthesis and functional material discovery. So there are 5 thrusts in the institute. The first 2 all focus on the development of AI tools for synthesis planning, catalyst selection, and optimization. So we are not limited to enzymes. We also look at chemical catalysis as well. And our idea is to combine, chemical catalysis with enzyme catalysis, right, to achieve more efficient synthetic routes. And then in thrust 3 and 4, we try to demonstrate those AI tools for different applications. In thrust 3, we choose 3 small molecules. 2 of them are drugs. The third one is a molecule used in polymer processing. And in thrust 4, it is organophotovoltaic material. We try to improve the efficiency and the stability. And recently, we just got the center renewed for another 5 years. And so here are the key deliverables in the center. So basically, we try to develop all the AI tools for molecular synthesis, and we call it alpha synthesis. Basically, the AI tools could be used to mine literature or existing database, and also the data that we will generate using like BioFoundry or similar systems for chemical synthesis as well. And then we have this database that contains the chemical reactions, enzymatic reactions, or the mutants, for different proteins. And then we leverage this database, try to develop new AI tools for synthesis planning, catalyst selection, and optimization. And another key deliverable is the AI-powered closed loop for the engineering of catalyst. And here we used 4 kind of case studies. One is related to Suzuki coupling reaction, try to optimize the yield of the reaction. And the other 2 are related to enzyme engineering. And also the 4th case study is organophotovoltaic materials. We try to improve the stability and efficiency. So here I just highlight a few AI tools that we developed. One is this AI tool for predicting enzyme function. Mainly is the enzyme commission number that will indicate what reaction the enzyme catalyze or what are the substrate it will use. And many of you, of course, are familiar with this AlphaFold, which is used to predict the protein structure from sequence. And that was developed by DeepMind a few years ago. And they also now is widely used in protein science and engineering fields. While with our tool, we now can really know the enzymatic reaction for the proteins. And also, more recently, we developed another AI to predict the enzyme substrate. Because even we know the enzyme reactivity, like what a reaction to catalyze, but there are so many small molecules that could be used as a substrate. So in many cases, we don't know which one is the best. So that's why we also developed a follow-up tool to predict the enzyme substrate. So we can rank, the molecules using this AI tool and predict which one will be the best, for the particular enzyme. And in addition to those AI, tools for predicting enzyme functions, we also developed the AI tools for enzyme engineering as well. So almost 5 years ago, we developed the AI to predict mutations that can improve protein function, particularly are those high order mutations, meaning that the combination of several mutations that can improve the protein function. And also more recently, we combined the AI with the robotics to achieve autonomous enzyme engineering. So in this new paradigm, we use the protein large language models to predict mutations that can improve protein function, and then use the robotics, the biofoundry, to perform all the experiments, including site derivatigenesis, characterization, right? And then based on the data we generated, we used the AI model to predict the combinations mutations that can further improve the protein function. And also we developed a chatbot, to help the researchers to design experiments as well. And as shown here, we used two enzymes as a test case. One is a methyltransferase that is used as a catalyst. The other one is a phytase that is used as an animal feed. And so we performed 4 rounds of kind of a direct evolution. So in the first round, we used the protein large language model to predict more like 180 mutants that can potentially improve the protein function. And then we made all of them and quantified their activities. And we found that indeed, ranging from like 20% to 50% of the mutants showed improvement. This is a much higher success rate than random mutagenesis, right? That people use in classical direct evolution experience. And then in the second round, we use an AI model to predict the combinations. So we construct a much smaller library of variants, only like 96 of them. And in the subsequent round, we all construct no more than 96 variants. So in total, we constructed and characterized less than 500 mutants, and yet we can significantly improve the enzyme function, right? As I shown here. So now, of course, we also try to make our technology broadly available. So with the funding from NSF last year, we established this national infrastructure called Biofoundry, in which we try to develop a cloud biofoundry that seamlessly integrates synthetic biology, AI, and automation to democratize basic and applied biological research. So we have an in-house research program in which we will continue to develop AI tools and automated workflows for enzyme engineering and cellular engineering. But also we have a user program in which we want to engage the broader research community. So we will solicit the proposals and we will fund those proposals and encourage them to use our facility and all the tools that we develop. Or, we could collaborate to develop new tools as well. And also, we launched another NSF center focusing on Biofoundry, but mainly through collaboration with partners in other countries. As I mentioned in the beginning, now there are more than 40 academic biofoundries, right, in the world. So we particularly partner with the biofoundry from UK, Japan, Korea, and Finland. And our goal is to develop the standards and the matrix to make the biofoundry more reliable and scalable. Currently, biofoundries all use different equipment, different workflow, different tools, different ways to manage the data, right? So it's very hard to share the workflows. So we basically try to create some standards among the community. So in summary, what I talk about today is that we developed a new strategy for enzyme catalysis by combining photocatalysis with enzyme catalysis to create photoenzymes with new to nature reactivities. And I showed a few type of reactions that can be catalyzed by these new class of artificial enzymes. And also, I talk about the AI Institute that I lead to develop new AI tools for molecular discovery and synthesis. And also, I introduced a few AI tools for enzyme function prediction and enzyme engineering. Right? And we're developing many other AI tools as well. And particularly, I think the take-home message is the AI-driven autonomous experimentation. I believe is a new kind of a research paradigm, and this also can be applied for chemical synthesis and material discovery and synthesis as well. So finally, I would like to thank the students and postdocs who did most of the work. Many of them have left my group already, but particularly I would like to thank DOE, which provides most of the funding for us to develop the capabilities for the biofoundry, and then NSF in recent years for the development of all the AI tools for molecular synthesis and discovery. So thank you for your attention, and I'll be happy to take questions.
Great. Thank you to all our speakers. We have time for some Q&A, but I just want to start by thanking everybody for some really insightful and interesting talks. I think as someone with a background in organic chemistry, it's wonderful to see how the beautiful reactivity that's being obtained through biocatalysis. I come from a background where we're used to sort of engineering very clunky enzymes iteratively, and the power of being able to just screen, hundreds of enzymes for desired activity very rapidly is just, it's fantastic. So we have a number of questions in the chat. We are coming to the end of the allotted time, so we'll run for about 15 minutes in the Q&A. I'll try and sort of synthesize the questions into broad themes so that as many people as possible can have their questions answered. One of the themes that has come up from a few people in the questions for the panel as a whole is about scale, about the challenges of large scale. So for example, Emily asked about if there are particular challenges in scaling bioelectric catalysis versus conventional biocatalysis. We have questions about the impact of AI-driven enzyme optimization on industrial scale biocatalysis. So I wonder if anybody from the panel could speak to particular challenges and opportunities in the industrial collaboration and large scale biocatalysis.
I think Emily's question is excellent. So if you look at the biocatalysis community, there's a lot of effort in sort of bioreactor designs, automation of sort of biofoundries, as we saw with Humans Talk, and so I think technology there is evolving, but at the same time we're just starting to use bioelectric catalysis for kind of electrosynthesis applications, and so there aren't bioreactors that have really been designed to have electrodes in them. Maybe electrodes from a sensor perspective, but not electrodes in terms of the electrosynthesis that would go on there. And so I think it's a great opportunity. We've been talking to a lot of people about sort of reactor engineering recently, because an electrochemical biofoundry would have to be something different, and it's definitely an area we're interested in going in.
Thanks. I wonder if Professor Zhu in particular, you mentioned that the institute you're at does a lot of collaboration with industrial partners. I mean, I wonder if you could speak generally about how that's worked, and if there's any advice you would give to researchers in academia in the audience who are interested in working on this scale and getting collaborations with industry established. Are there any particular challenges, whether they're technological or social or cultural, that you've encountered there that you can speak to?
Yeah, for the collaboration with the industry, I think the important thing is establishing trust between the collaborators. So also need use a proper mechanism to do that. So we usually establish a long-term collaboration with partners. If we usually do not like only one transfer. So we started from the industry question together and then work together and then push this technology to the commercialization together. So it's kind of difficult, yeah. It's not like a published paper.
Yes, it's a whole other order of magnitude than publishing a paper.
Yeah.
I mean, there is also quite a specific question here, I think, about the role of AI on industrial scale reactions. So I'll read the question out because it's along this theme, but I think it's quite timely. So this is from a researcher at Zhengzhou University who asks what the key technical bottlenecks facing the application of AI-driven enzyme optimization in industrial scale biocatalytic reactions, and what effective breakthroughs have been formed in existing research. I mean, maybe it's a question for Professor Zhao, perhaps, as you talked quite a lot about AI in your talk and your work.
Yeah, I would say currently there's a renaissance in enzyme engineering. Really is because of the development of AI tools, right? Currently, there are many startups now focusing on using AIs for protein engineering, and also in industry like the existing companies as well. And I think the reason is because we have those protein large language models that really enable the prediction of protein functions and also engineering. So there are many tools now we can use to accelerate the enzyme engineering effort, and particularly those robotics also help the actual experience greatly as well. So I think there's a lot of activities in this field, and I cannot point out any real success yet. But this field is also very new. I'm sure in the next few years there will be many kind of industrial successful examples that really use the AI tools for enzyme engineering and also scale up.
Certainly, as an editor, we get a lot of research in the last 5 years using large language models and AI in various applications, and I think enzyme engineering, protein design, and also materials discovery are areas where it seems to be having a real impact and real success, versus some other areas where maybe it's a little more hype driven. I mean, there's a very specific question for you, Professor Zhao, about your AI driven pathway optimization workflow. You've been asked, how do you determine the appropriate number of design and test iterations, and specifically when to decide to stop the cycle? And then following on from that, how do you verify the final result is truly the best producer, not a local optimum within the design space?
Yeah, I mean, this is a good question. This is also a question for any direct evolution experience, right? In reality, is that we stop the one, the students run out of time, but now we have the robotics, so we can continue in principle, right? But so then the stop point is really that once we meet the target performance matrix, right? So we know that how much improvement we have to make in the activity or stability, and then we just use the AI-driven autonomous experimentation platform, do it iteratively, and see that whether we can meet them or not. But actually, recently I was asked, what happens if we run the experiment like 1,000 times or even more, right? What will happen? Because I'm curious about that, So we probably will do that in the near future.
The scale is very impressive. While we're on the subject of technical questions and specifics, we have I think the first question we got actually in the chat comes from Alice, who has a broad methodological question, which maybe for anyone on the panel, about how to identify effective high throughput workflows for identifying enzymes with the desired function, and she's particularly interested in thinking about how to prioritize candidates that can translate from, say, a bacterial model to a mammalian model. Is that something that anybody in the panel can speak to?
Yeah, we are recently developing an enzyme retrieval AI tool. So basically, and this is a very good question because there are so many enzymes out there, right? How do you know the enzyme you use is the best out there, right? Now, we're not talking about the enzyme engineering because with the enzyme engineering, you can always improve the protein function. But just pick the starting point, right? How can we find the best enzyme for any experiment? So I think we really try to develop some AI tools to first one to identify the enzymes for a given reaction. But then the more important thing is also the enzyme property prediction tools we develop. And we now can predict the stability, the catalytic efficiency, the solubility, all kinds of properties, right? And then you can have a kind of a prioritized list of enzymes for testing. And then that will help you to find the best enzyme very quickly. Now, those AI tools not 100% accurate, but it can at least help you to narrow down the targets. And I think this kind of a strategy will be increasingly used in the future.
When you refer to AI models in this context, are you referring to sort of just machine learning in general, or are you specifically thinking about the current wave of generative AI, large language models?
Not a generative AI. It's those AI tools that, like I showed 2 examples, right? We developed the AI tool for predicting the EC number, also the subspecificity, but there are also many AI tools for predicting the KM and the solubility, stability of the enzymes, right? So now you just need to screen all the enzymes out there, and then you can identify the one with the best properties out there, and then you can use that one to do further engineering or further testing.
Thank you. And for the last couple of minutes, we have one final question for Professor Zhu. About the construction of chiral centers and how your team balances precision of enzyme structure analysis with the efficiency of engineering strategies in the context of constructing chiral centers for a desired product.
Oh, this is a good question, but it's very hard to answer this question. Yeah. So usually we just like Huimin said, so we use computation to predict, and then use experimental to verify, and then do analysis again, and just do it several rounds, and then get the target enzyme.
Well, like I said, I think we're a little bit over time, so I think we'll have to wrap up here, unfortunately. But thank you to all of our panelists for joining us and for sharing such exciting research. And thank you to everybody for listening and for sharing your questions. There will be a recording of this email to all registrants on Monday, and it will be online on cell.com next week. So if any of your colleagues missed out and are really excited to follow up after the fact and hear about the exciting work we've discussed today, then they'll be able to do so. So again, thank you to all of our panelists, and thank you everybody for attending.
Okay, thank you very much.
Thank you.
Bye.